Welcome to the Gramene webinar series. My name is Kapeel Chougule. I'm a computational science developer at Cold Spring Harbor Laboratory. So today we'll be doing a Gramene webinar on transforming genomic coordinates between plant reference assemblies. So I'll be talking to you about the assembly converter tool that we have on the Gramene browser. So before we get into the details of the webinar, a few technical tips. So if you, during the course of the webinar, if you notice any video halting, so please select a lower quality stream. You can find it on the drop down menu on the top left of the window, video window. You can also see a full screen video by clicking on the button with 4 arrows at the top right of the video window. And if you have any questions, please feel free to type in at the chat panel that you see below the video window. You can alert us for any issues that you might face by typing it on the chat panel. So with that, we'll begin with the Gramene webinar. So Gramene hosts the assembly conversion tool on the browser. So what it basically does, it converts genome coordinates between assemblies. So you have like many versions of a particular assembly. So this kind of tool helps you in doing direct comparison between different versions. It helps in data integration and visualization, and it also facilitates for doing meta-analysis. Then the question comes, why do we want to use such a tool? So as I said before, that this kind of tool helps you in converting coordinates from older assemblies to a newer one. And we know that nowadays there are a lot of genome assemblies that are being updated, and so it becomes very vital to do the conversions from one version to the other. So Gramene, the assembly converter tool that Gramene hosts, the underlying tool for that is CrossMap. So it's an open source tool. You can find the citation below on the slide. So the way CrossMap works is it basically determines correspondence between the two genome assemblies using the UCSC chain file. So the UCSC chain file is nothing but a pairwise alignment between two genomes. So and then the genome intervals it stores in a interval tree data structure. So if you want to find an overlap for all the intervals, you can basically navigate through the interval tree data structure. So what CrossMap does is it basically queries each of your query from the input file. So here you bring in input files which can be in different file formats. That could be a BAM file, a SAM, or a VCF file. So CrossMap basically takes each entry from that and it remaps to the target assembly, and then it queries through the interval tree data to convert the coordinates from an old assembly to a new assembly. So so CrossMap is just one of the tools. So there are many other conversion tools, each with its pros and cons. We have like UCSC liftover tool that supports like only bed as an input. And then you have the NCBI remap. It has many other formats that you can use as an input. Galaxy, the platform, it also hosts a few of the conversion. And then you have the Pi liftover. It basically does like a point conversion. So there are many other options. But CrossMap has a distinct advantage that it can host many different file formats, and also it can host like a BAM file and others, which are pretty useful. So on the Gramene browser, so we have like the many different suite of tools. One of them is the assembly converter. So you can basically find it from the menu bar by clicking on the tools, and you can see the link over there. And then you have the assembly converter tool. When you click on that, it will bring up the job submission page. So here you see many different options. So we have the species that you want to select for doing the assembly conversion. Then you can choose the assembly mapping. So we have assembly mappings from different versions of the assembly. And then you can give an input for the a name for your job and select the file format. And there are 3 different ways you can actually bring in your data set. You can either paste in the contents of your input file directly in the text box, or you can upload your file through the browser, or you can provide an external HTTP link directly into the job submission page. So once you click on that, you'll probably see the progress of your submitted job. You can see if it's queued, if it's done, or if it's failed. You can also resubmit a particular job. So that I'll be showing you in the next few slides during the demo. This is how the web page for the assembly conversion tool looks like. And we support a few species, a total of 11 of them currently. We also have the chain files that are required for assembly conversion. So if you plan to do conversion using standalone tools, you can very well use the chain files to do that. For now, we support 11 species. So for this webinar, I'll be basically talking about 2 file formats, the GFF or the GTF ones and the BED ones. So first I'll talk to you about how we convert a GFF file format. So a GFF, for those of you who are not aware of this format, so it's basically a gene feature format. It basically describes a gene structure. So it's a plain TXT file. A sample one is shown below, where each line each line of the GFF, it describes the feature, the genomic feature, like on what chromosome it is, the coordinates and what strand the feature is on, and also certain other metadata in the last column. So cross map, the tool itself, it will take each feature and it processes separately and independently. And then you can bring in the GFF file as a plain text, compressed plain text, or you can even use a URL. So all those input types are supported. And just to note, it only chromosome and genome coordinates are updated with this feature format. So I'll be explaining the demo with this input. The next one is the BED file format. So the BED, it's also a browser extensible data file format. It's a tab-delimited one. Again, it describes genome regions and gene annotations. So a BED file can comprise like a minimum of 3 columns, and it can extend up to 12 columns. But just to get it working, we can just specify the chromosome and the start and end coordinates. So with the cross map, the regions of the old assembly which map to the multiple locations on the new assembly will be split. So you can expect certain genes which from the old in the new assembly be split, which were just as a single gene in the old assembly. Then you can also bring in a compressed file format for this particular input. So the tool itself has certain limitations, so I'll be listing out a few of them. Firstly, since it's a web-based tool, you have a size limit of 50 MB. So if you have a bigger file, it would be good that you run it standalone by downloading the chain file and using cross-map. Then we also have to take into consideration the chromosome numbers, how the IDs are specified. You'll probably have to strip off the prefix if you have ID, like for example, I've mentioned the O-glabrima chromosome 1. You need to strip off the prefix to just the numerical number of the chromosome. So the reason for that is that's how the genome assembly is integrated into the browser. So if you have like a scaffold ID or something, then you need to match according to what you have on the browser. So that's one of the considerations while you're running this tool on the genome browser. So then and currently the assembly converter tool doesn't support VCF BAM big wig format yet. So that's another thing that will be available in the future releases. So with that, I'll switch over to a demo. I'll basically describe how you can run an analysis using a GFF input and a bed input. So basically, when you come to the Gramene page, you click on the Tools button. You'll see the list of the tools. You click on the Assembly Converter tool. You'll see the options where you have the species. For now example, we'll select the Oryza japonica. We'll convert from IRGSP1 to MSU6. Just give a name for the input for the job. Select the file format and choose the file. So here we'll choose a small test GFF file for the Sativa one. Click on Run. So you'll see the job submission, you'll see the status of it. You can save it, edit it, or delete it. And then you'll see it refreshes for every after 10 seconds. And once it's done, you click on the download results. Similarly, you can do this using a bed input format. We'll use the same species and the same mapping. Again, we'll use the bed format. So you can see it's queued, and you have also other tools, the web-based tools that you can navigate through if you want to search through some of the data. And then once it's done, you click on the download results. So that's basically how you run the assembly conversion tool. So for the results, so if you can see the input that I used for the demo, it basically was IRGSP coordinate GFF file. So this was like a small test file. So now after running the assembly conversion tool, this got converted to the MSU6 coordinates. So the column 4 and 5 represents the genome feature coordinates. So they are converted to the MSU6 coordinates. Similarly, you'll see the bed input. So here the coordinates are on chromosome on column 2 and 3. So they too get converted to MSU6 coordinates. So it's a very simple tool to use. It's you need to just take into account what format that you're bringing in, and also make certain modifications. So if you have seen that the first column here, it just has the numerical number for the chromosome. So make sure you map it accordingly. So that was a short demo on how you run the assembly conversion tool. So we have many other webinars on topic specific related to plant, like Arabidopsis, rice and maize. So if you follow our Gramene channel database on YouTube, you'll find all about 21 recorded webinars, and also we have a mailing list of about 1,000 plant researchers. So you can see, and this webinar will be also available on the YouTube channel. So there are various ways to reach us. You can email us at webinars@gramene.org, or you can use a contact form from the website. And also you can, if you're active on social media, please do contact us from Facebook or Twitter. And lastly, I'd like to thank the folks working on the Gramene project. I'm part of the Ware Lab group, and the funding agency, the National Science Foundation. And the people highlighted in green, they work on the genome component of the Gramene project. So thank you for listening, and if you have any questions, I'll be happy to answer some of them.